36 years old. Zero warm-up. Ball delivered to his feet at 65 miles per hour. And within 200 milliseconds less time than it takes you to blink, Cristiano Ronaldo has already decided where he's shooting. Which foot? At what angle? With how much spin? The ball is in the net before your visual cortex has finished processing what you just saw. That is not reflexes. That is not experience. That is neuromuscular architecture operating at a level most human beings will never reach. And today, we are going to examine exactly what that architecture looks like, down to the individual protein filament. Every coach will tell you the same thing. He trained 10,000 hours. He stayed after practice. He wanted it more. And here is why that explanation, while not wrong, is almost entirely useless. Because there are thousands of players on this planet who trained 10,000 hours. Who stayed after every session. Who wanted it with every cell in their body. And none of them can do what Ronaldo does at 36. The hard work exclamation mistakes the stimulus for the adaptation. It describes what went in but says nothing about what came out. What came out, in Ronaldo's case, is a neuromuscular system that has been so precisely and specifically remodeled at a molecular level that it belongs in a different physiological category from a recreational athlete. So the real question, the one nobody in sports commentary ever asks, is this: what exactly changed inside his body? And can it happen inside yours? What we are going to do in this video is examine 3 separate but interconnected biological systems and show you the exact molecular mechanism operating inside each one. The research comes from multiple institutions. We'll draw on work published by Dr. Perogard at the University of Southern Denmark, who has spent 3 decades studying elite neuromuscular adaptations. We'll reference a 2019 study published in the Journal of Applied Physiology, examining type 2 muscle fiber hypertrophy in professional footballers. And we'll look at neurological research out of the Danish National Research Foundation's Center of Excellence for Muscle Function. This is not sports science generalism. This is the specific, molecular, mechanistic story of why Cristiano Ronaldo's muscles fire differently from yours and the biology your coach never had time to explain. To understand what makes an elite footballer's muscles extraordinary, we have to start not at the muscle, not at the fiber, but at the molecular machine inside the fiber, the sarcomere. Think of a sarcomere like a biological piston engine, except instead of pistons driven by combustion, this engine is driven by chemistry. Inside every sarcomere are 2 types of protein filaments: thick filaments made of a protein called myosin, and thin filaments made of a protein called actin. When your nervous system fires a signal telling a muscle to contract, myosin heads tiny molecular. Motors reach out and grab onto actin filaments, pull them inward, and release. This is called the cross-bridge cycle. And it is the fundamental mechanical event underlying every movement your body has ever made. Now here is where it gets important. The speed at which this cross-bridge cycle operates is not the same in every human being. It varies. And it varies for a specific reason. Human skeletal muscle contains 2 primary types of fibers. Type 1, which are slow-twitch fatigue-resistant, efficient, designed for endurance, and type 2, which are fast-twitch. Fast-twitch fibers contain a different isoform of the myosin protein. Different isoform means different molecular shape. Different molecular shape means different cross-bridge cycling speed. Type 2X fibers, the fastest subtype of fast-twitch, operate their cross-bridge cycle at approximately 3 to 5 times the speed of type 1 fibers. The average untrained adult has a fiber-type ratio of approximately 50% type 1-2. 50% type 2. Elite sprinters can show type 2 composition above 70%. And in professional footballers, specifically those in the mold of Ronaldo, who combine explosive acceleration with technical precision, research has found something more nuanced and more interesting than simple fiber percentage. A 2016 study by Ogard and colleagues examined the muscle biopsies of elite Danish football players and found that years of high-intensity explosive training did not simply increase the proportion soft, fast twitch fibers. It changed the internal architecture of those fibers. But this is where it gets strange. Because the mechanism that makes Ronaldo's muscles fire faster is not actually in the muscle at all. It's in the wire that controls it. The motor neuron. Every voluntary muscle contraction begins with an electrical signal propagating down a motor. Neuron, a nerve cell whose axon can stretch from your spinal cord all the way to a muscle in your foot. The speed at which that signal travels is called nerve conduction velocity. In a typical adult, motor nerve conduction velocity sits between 50 and 70 meters per second. But here is the critical variable. What determines how fast a muscle fiber responds is not just the speed of the nerve signal. It is the synchronization of that signal across multiple motor units firing simultaneously. A motor unit is one motor neuron and all the muscle fibers it controls. In a fine motor task writing your name, you might recruit a motor unit controlling 10 fibers. In an explosive jump, a Ronaldo leap at the far post, you are recruiting motor units controlling hundreds of fibers simultaneously. The question is, how synchronized is that recruitment? This is where elite athletes diverge from everyone else at a level that is completely invisible from the outside. Let's put this system under match conditions and trace what actually happens. Ground contact time. When you or I run at moderate pace, our foot contacts the ground for approximately 250 to 300 milliseconds per step. Elite sprinters reduce this to around 80 to 100 milliseconds. Ronaldo, during his top speed sprinting phases captured on motion analysis systems at clubs like Manchester United and Juventus, has been measured at ground contact times approaching 85 milliseconds. 85 milliseconds! That's roughly 1/12 of a second. In that window, the entire mechanical sequence of loading the tendon, activating the muscle, generating force, and pushing off must complete. If any component in this chain is slow, if the neural signal takes too long, if motor unit recruitment is poorly synchronized, if the muscle's cross-bridge cycling speed is insufficient, the window closes and the force goes to waste. The mechanism that enables this is called the stretch shortening cycle, the SSC. When the foot lands, the muscle tendon unit is rapidly stretched while under load. This stretch stores elastic potential energy in the tendon, primarily the Achilles tendon, like a compressed spring. Immediately after, and this timing is critical, it must happen within a precise window, the muscle contracts eccentrically, then concentrically, releasing that stored energy on top of the active muscular force. Research from Professor Paavo Komi at the University of Jyväskylä in Finland, widely considered the world's leading authority on the SSC, showed that in elite athletes, this elastic energy recovery can account for up to 50% of the total mechanical output during a sprint stride. In other words, half the propulsive force isn't being generated fresh with each step. It's being recycled from the previous step. The body is functioning like a biological piston engine with an energy recovery system built into the drivetrain. Here's what nobody tells you. This elastic energy recovery system only works if the muscles are stiff enough, and that stiffness is actively regulated by the nervous system in real time. Muscle tendon stiffness is regulated by two competing neuroreflexes: the stretch reflex, which increases stiffness, and the Golgi tendon organ reflex, which decreases it as a protective mechanism against tearing. In an untrained person, these reflexes operate conservatively. The Golgi tendon organ fires early and often, reducing force output to protect the tissue. In an elite footballer, years of specific training have recalibrated these thresholds. The inhibitory reflex from the Golgi tendon organ fires later, allowing the muscle tendon unit to operate at higher stiffness levels for longer. Research published in the European Journal of Applied Physiology by Maffioletti and colleagues found that professional footballers demonstrated significantly higher tendon stiffness measured via ultrasound elastography compared to age-matched recreational athletes, and that this stiffness correlated directly with jump height and sprint speed. Now, here is the number that should stop you mid-thought. In a study measuring motor unit discharge rates, the firing frequency of individual motor neurons during explosive contractions, elite sprinters showed peak discharge rates of 150 to 200 impulses per second. Untrained adults typically reach 60 to 80 impulses per second. That is not a marginal performance advantage. That is a fundamental difference in how the nervous system is operating. And this leads us to what is arguably the most sophisticated aspect of Ronaldo's neuromuscular biology, something that doesn't get discussed in sports media at all, because it's not visible. the predictive motor program. A motor program is a pre-planned sequence of muscle activations that the brain executes before sensory feedback can arrive to correct it. At 85 milliseconds of ground contact time, there is no possibility of real-time correction. The brain must predict what the body will need and fire the motor sequence before it has confirmation that the prediction was correct. In novice athletes, this predictive system is poorly calibrated. The motor program fires, feedback arrives, corrections are made, but those corrections take time. 40 to 100 milliseconds depending on the complexity of the correction. At elite speeds, that latency makes you a fraction too slow on every single step. In Ronaldo, after 2 decades of extremely specific neuromuscular training, the predictive motor programs are so precisely calibrated that the latency between intention and outcome is compressed to a degree that researchers at the Karolinska Institute in Sweden have described as approaching the theoretical minimum for voluntary motor control in the human nervous system. So, how did this system get built? Because none of what we just described is genetic fate. These are adaptations, and adaptations require specific stimuli delivered over specific time frames. The story begins at Sporting CP's academy, where Ronaldo arrived at age 12 from Madeira. What the coaching staff observed almost immediately was not extraordinary skill, it was extraordinary willingness to perform repetitive explosive movements. Sprints, jumps, change of direction drills, not for fitness, for technical improvement. But the biological side effect of that technical repetition was exactly the neuromuscular stimulus required to drive the adaptations we've been describing. Myelination. The myelin sheath is a fatty insulating layer wrapped around axons nerve fibers that dramatically increases the speed of electrical signal transmission. Each layer of myelin can increase nerve conduction velocity, and critically, unlike muscle hypertrophy, which can be stimulated in months, myelination is a slow, long-term process. Research from neurobiologist Dr. George Bartsakis at UCLA found that white matter myelination in motor pathways continues to develop and optimize throughout adolescence and into early adulthood, with peak optimization occurring somewhere between the ages of 25 and 35, which means that the explosive neuromuscular training Ronaldo began at age 12 was not just building muscles, it was building neural infrastructure that would reach its peak refinement precisely when he was in the middle of his professional career. The 10-year myelination window aligned almost perfectly with his development arc. That is not luck. That is an extraordinary coincidence of biology and biography. And this next part is what the data actually shows, because genetics does play a role here, but not the role you've been told. The ACTN3 gene encodes a protein called alpha-actinin 3, found exclusively in fast-twitch type 2 muscle fibers. There is a well-studied variant of this gene, the R577X polymorphism, where the X allele produces a non-functional version of the protein. Athletes who are RR genotype 2 functional copies consistently outperform XX genotype athletes in explosive power tasks. Studies of elite sprint and power athletes by Dr. Nurainan and colleagues at the Australian Institute of Sport found RR genotype prevalence of over 80% in elite sprinters compared to approximately 50% in the general population. But here's the critical point, and it's the point that most popular science gets wrong. The ACTN3 genotype does not determine whether you become elite. It influences the ceiling of certain explosive adaptations. The research consistently shows that XX genotype athletes can still develop exceptional speed and power, they simply require longer training, timelines, and more specifically targeted stimulus to achieve comparable adaptations through compensatory mechanisms involving other fast-twitch proteins. And this is where it stops being about Ronaldo and starts being about you. What we have described across the last 10 minutes resolves to a single mechanical principle: the neural specificity law of motor adaptation. The neuromuscular system adapts with extraordinary precision to the exact mechanical demands placed upon it repeatedly. And those adaptations are layered across multiple time scales simultaneously. Short-term, within weeks, motor unit synchronization improves. The nervous system learns to recruit more fibers at the same moment increasing peak force output without adding any new tissue. This is why strength gains in the first 6 to 8 weeks of a new training program almost entirely represent neural adaptation, not muscle growth. Research from the late Dr. Digby Sale at McMaster University in Canada documented neural efficiency gains of up to 30% within the first 8 weeks of resistance training before any significant hypertrophy occurred. Medium term, within months, the cross-bridge cycling mechanics begin to optimize. Type 2A fibers, intermediate fast-twitch fibers, shift their myosin heavy chain isoform expression. Studies using single-fiber electrophoresis have shown that type 2A fibers in trained athletes contain a higher proportion of the faster MHC 2A isoform compared to untrained. Controls. The molecular motor gets faster. Long-term, across years and decades. Myelination deepens. Tendon stiffness increases, and the predictive motor programs achieve the calibration we described in Ronaldo's case. This law does not apply only to athletes. Every elite performer whose skill requires precise, rapid motor execution is operating under the same principle. Neuroscientist Dr. Arne Dietrich at the American University of Beirut has published work showing that jazz pianists who have practiced improvisation for 20 or more years show motor cortex activity patterns during performance that are structurally indistinguishable from deep motor habit. The cortical overhead of thinking about hand placement has been almost entirely eliminated, with execution being controlled by subcortical and cerebellar circuits that operate at speeds the prefrontal cortex simply cannot match. And in Formula One racing, telemetry data from teams including Mercedes-AMG Petronas has been used to show that elite drivers begin making steering corrections before consciously processing the sensory information that prompted the correction, a phenomenon explained entirely by the predictive motor programming mechanism we described earlier. The mechanism is universal. The time scale of development is universal. What changes is only the specific motor pattern being optimized. So, what does all of this mean for you, not as an aspiring elite athlete, but as someone who now understands the actual mechanism? It means that when you begin a new physical skill, a sport, a martial art, a dance form, a musical instrument, and you feel clumsy and slow for the first several weeks, you are not experiencing a lack of talent. You are experiencing the early stage of neural recalibration. Your motor units are learning synchronization patterns they have never been asked to execute before. The sensation of clumsiness is the sensation of an unsynchronized motor orchestra trying to play a piece of music for the first time. It means that when you feel a sudden, inexplicable jump in performance after a period where nothing seemed to be changing, the familiar breakthrough experience, you are almost certainly experiencing a threshold in myelination or motor unit synchronization being crossed. The adaptation was happening the entire time. It simply wasn't expressing itself as visible performance because it hadn't yet crossed the functional threshold. It means that the specificity of your stimulus matters far more than its volume. Ronaldo did not become neurologically elite by running long miles. He became neurologically elite by performing explosive, precisely timed movements repeatedly, from an early age, across a long enough timeline for myelination to compound. You can replicate the mechanism with vastly different content. The biology doesn't care whether it's a football pitch or a squash court or a tennis baseline. What it cares about is the precision and explosive intent of the movement pattern. And this is what the data actually shows about elite performance longevity, because the most remarkable thing about Ronaldo at 36, 37, 38 is not that he is still fast. It is that the adaptations we've described, particularly the myelination and the tendon architecture, are among the most durable biological structures the human body produces. They do not atrophy at the rate that muscle mass does. They are, in a very real biological sense, the most permanent physical record of a life's worth of training. The myelin doesn't forget. The tendon architecture doesn't forget. The motor programs don't forget. They can be suppressed by disuse, but the structural substrate remains. Which is why the most accurately named thing in all of sports science is not a training method or a recovery protocol. It is the most common observation made about every athlete who reaches elite level and then takes years away from their sport before returning. They call it muscle memory. And now you know it's not in the muscle at all. There is one part of this story we haven't told, and it may be the most important part. Everything we described today, the motor unit synchronization, the myelination, the SSC mechanics, the predictive motor programs, all of it operates downstream of one system. A system that Ronaldo has been reported by his own coaching staff, his teammates and sports scientists working with him at Juventus to have optimized to a degree that borders on the obsessive. That system is sleep, not recovery in general. Sleep specifically. And the reason sleep is not just a recovery tool, but the actual biological mechanism through which motor learning occurs. Consolidates the reason the myelination we described quite literally does not happen without it is a story about a process called synaptic homeostasis that researchers at the University of Wisconsin-Madison have only recently begun to fully map, if you want to understand why. Ronaldo reportedly sleeps in 5 90-minute cycles rather than one continuous block, and why the science behind that specific protocol is more sophisticated than any training method we discussed today. That is the next video. Subscribe now, because the next video answers the question this one raises. If the neural adaptations we described are the hardware, what is the operating system that builds and maintains them? The answer is not what the sports world thinks it is. And while you're waiting, the video that pairs most directly with what we cover today is linked on your screen right now. It covers the biology of tendon adaptation specifically, why the Achilles tendon in elite jumpers stores and returns energy at efficiencies that approach the theoretical limits of biological tissue, and what the collagen cross-linking mechanism behind that efficiency actually looks like at the molecular level. Watch that one next.